right here a little bit in detail. 2 kidneys we have, and then this is the ureter. People often does mistake on ureter and urethra. Don't do that mistake. Ureter is the one that brings the urine from kidney to the bladder for temporarily store them, and urethra that takes the urine out from the bladder to the restroom, like in the environment. Okay, 2 kidneys. We mentioned all these things. That's why I'm going a little bit fast here. 2 kidneys are not exactly aligned like a parallel. The right kidney is slightly lower. This picture is from the back, so this is the right kidney. So it's a posterior view. So the right kidney is slightly lower. What does that mean? That means this is slightly inferior, that's slightly superior. That's the word they use. Okay. Again, if you donate, if you get a chance to donate a kidney, or if you have to donate a kidney, do not get afraid because your one kidney, it is well proven that one kidney can do the regular job which 2 kidney can. And I'm just going to mention this because I'm really proud of this fact, because one of my Suffolk County students in Grand Campus, he is actually the spokesperson of the kidney donation unit in Long Island. He was a donator of his kidney to his best friend. I really have a lot of respect for him. Okay. He was my student in the NP class. So these are the 2 kidneys. Again, inside of the kidney, we learned that. I told you to study from here because these words are written on the side. So when you prepare yourself for the lab identification, you already know the intricate structure of the kidney. We talked about this, the blood vessel service, very important for your lecture and the lab. Please read them again. These are the 2 sets of nephron, not neuron, nephron. Nephron is the unit that we studied already, the renal corpuscle. In this picture, if you go back, this portion one more time, this portion on the end, that's called cortex, and where the pyramids are, this is called medulla. Okay, so this portion is called renal cortex, the top part. The inside part is renal medulla. The cortex has 85% of our nephron population. So most nephron, or 85% of the nephrons, are right here. They are called cortical nephron. Simple word based on the where they're located, meaning they're located in cortex. That's why the name cortical nephron. And then there are 15% nephron. What they are, they can begin here in the cortex area. If you think these blood vessels divide the cortex and the medulla, their loop is in the medulla. There are few differences between these 2 nephron, how they work. In your undergraduate level, you do not need to know that difference very detailed. So you should know that there are, I think I have a slide on that. I'm just going to skip some right here, that 85% nephrons are cortical nephrons. They're a little bit short nephron loops. 15% of all nephrons are medullary nephron or juxtamedullary nephron. The word juxta means near. So near medulla, they start near medulla. That's why the name. And there are very long nephron loops. And this nephron loop, that straight line right here, that peripheral vein and artery, that capillary, is called vesa recta. The word vesa means vessel, recta means parallel. So they're basically around the long nephron loop. These blood vessels are very long and straight compared to these capillaries. This capillary is like a network, like a bed of blood vessel, and they're like a lot of straight vessel. They're straight vessel. That's why the word vesa recta. Recta means parallel, you know, and vesa means vessel. So they're like, this is the loop, they're parallel to the loop. So these are all the nephrons. Now what they do is basically same thing. All of them, they have 2 parts, the renal corpuscle part and the tube part. The renal corpuscle is the one that's where the filtration takes place. The blood vessel going in, blood vessel coming out, both are artery, and then right here, the filtration takes place. So when you study this part, you need to know two very important things. You need to know about the blood vessels and you need to know about the inside layer. It's like a cup. So this is the inside layer and that's the outside layer. The gap between these two layers where these dark arrows are, that's what they call, that's what the space where the glomerular does the filtration. That's the filtration. So these cells of the inside layer, so inside layer is called visceral, the outside layer is called parietal. The word parietal is right here, visceral is right here. So these are the inside layer called visceral. This is the outside layer called parietal. Inside layer, that visceral layer is made with a cell, the special name podocyte. Podocyte, site means cell, podo, the word came from foot, like podiatrician, like a foot doctor, that's what, podo. So podo means, so podocyte is like a cell with a lot of feet. So the cells are like this. So when you have a lot of cells like this, all their feet together, they create the slit, the opening. That is the filtration. That is where the filtration takes place. That filter together, these podocytes and the blood capillary create the glomerular filter. So blood capillary right here, these are glomerular blood capillary. And the podocytes together, they create the membrane that we discussed already. So that glomerulus, where the filtration takes place. Now, who is going to come out? Not the blood cell, not the protein, the one that is small. So I put a slide right here. Hang on one second, right here. So this is the podocytes. These are the feet, like all these feet of the, it says foot process of podocytes. So there's a foot, each foot. So they have a gap between that. And also, this is the blood capillary network. So capillaries in the blood vessel chapter, I'm going to mention one extra thing here. Capillaries in the blood vessel chapter, we know there are three types of capillary. If you remember the name, continuous capillaries, fenestrated capillary, sinusoid capillaries, or sinuses. Fenestrated capillary, the capillary with more window, more space, that's this. The kidney, right here, these capillaries, blood capillaries are fenestrated. The word fenestra means window. So they have a little bit more space between cells. So in the molecules on the blood flow through these molecules, anything smaller, they will come out. So what can passing through? Water, electrolytes, glucose, amino acid, fatty acid, vitamins, urea, uric acid, creatinine, all these will come into the capsular space. What cannot come? Proteins, they can't come, or protein bound minerals and hormones, they cannot come. Large anions, they cannot come. Blood cell, they cannot come because they are more than these diameters. So they will return and continue in the blood. Now, out of these, which can come, we already talked about that. Body let this waste product like urea, uric acid, creatinine, along with some extra minerals, they will let it go. Anything extra, and then they will reabsorb 99% water. They will reabsorb 99% electrolytes, 100% glucose, 100% amino acid, 100% fatty acid, 100% vitamin. They want to absorb them. Extra, yeah, they let them go. So anything extra, they let them go. Because remember, everything has a threshold. So if you keep eating carbohydrate all the time at tons, obviously your urine may contain a little bit of carbohydrate after the meal. A little bit of glucose after the meal, but that's not abnormal because kidney has a limitation. So what you need to know in this remaining of the chapter, that what part does what job in the tubule, what part absorb these, what part absorb these, that we need to know. So if you go back right here, we have three parts: PCT, PCT, nephron loop or loop of Henle, and DCT, distal loop. So first, you need to know how they look like. So PCT, they're simple cuboidal cells with a lot of microvilli to increase the surface area, so that, because right here, majority of the absorption occurs. DCT, they also have simple cuboidal, but they don't have a microvilli. Not only that, in the DCT, which I mentioned the next slide, there is another cells with microvilli called intercalated cells that present. So DCT has two populations of cuboidal. Main population, like 90% of them are this, 99% of them are like this. And there are few that are this, intercalated cells, which they have microvilli, because functionally they are a little bit different, but that detail you don't have in undergraduate level. In the loop, we have descending, we have descending limb and we have ascending limb, but both the limb has a thick part and a thin part. Thick part is made with cuboidal cell, and the thin part is made mostly with simple squamous cells. Okay, the reason I wrote in the ascending thick segment has simple cuboidal / low columnar, because some book mentioned that those cuboidal cells are slightly taller, so some books say that is a low columnar cells. So that's their histology. That's how they look like. What they do, I'm coming on. But before we go there, just need to mention one extra thing here. If you pay attention here, afferent arteriole, the one that going in is wider, have a large diameter. Efferent arteriole, exiting, afferent going in, efferent comes out. They are narrower in diameter. So now this is two very interesting things here, very unique for kidney. Normally in a tissue and in an organ, we learn that the blood vessel that goes in, we call artery, that brings the blood to the tissue, and vein returns. Vein returns. In this case, artery going in, artery coming out. Very unique. An artery, we know, a little bit higher pressure vessel, so that helps, that actually helps to maintain the pressure here in the capillary bed is slightly higher than anywhere else in the body. Like anywhere else in the body means anywhere other capillary networks in the body. So normally in the capillary network, the blood pressure is around like 35, right, in the artery. Right here, because of that artery going in, artery coming out, because vein is a low pressure vessel, the blood pressure is slightly up. But there is another reason. Look at the size. The afferent is wider, efferent is smaller. Very interesting design. Why? Because more goes in, less comes out. So they keep the blood pressure slightly up. So the blood pressure here, that slight extra blood pressure help in the filtration process. So then add to it. I'm going to mention about the total net blood pressure a little bit later. So those are the two important stuff for renal corpuscle. This is the structure of the renal tubule. Again, continue, structure of the renal tubule. The flow we already learned, how the urine is flowing through. This is what I mentioned, that first the filtration, then the tubular reabsorption, then the tubular secretion, and then finally water retention, and then you produce the urine. This one I did a little bit take out that yellow circle. The reason is we do not need to know very detail of that. We need to more focus on here, here, and here. Secretion happens, that you should know, but how it happens, the detail, structure, function, what cell does it, you don't need to know. Okay. So this is how the urine forms. So urine form, first the filtration, glomerular filter filtered, then the tubular fluid is going through the tubule. Some absorb, some they let it go. And then finally it goes through the collecting duct, papillary duct, minor calyx, major calyx. I gave you that list, it's also on the slide, until the urinary bladder. And then urethra, put it in the environment. This is the pressure that I was talking about. So the blood hydrostatic pressure here is 60 approximately. Other capillary network is like 35. So that is helping the fluid, the molecules that is smaller along with lots of water come to the capsular space. So is that the pressure that working in your favor? Yes, but is that the only pressure? No, because there are two other pressure that working against that filtration. Against that filtration. What are those two pressure? One is called colloidal osmotic pressure. Another is capsular, like a hydrostatic pressure of the capsule, so the capsular pressure. Now we got to understand the two meaning of it. Like colloid, osmotic pressure is basically, colloid is a type of like a solution. So in the large molecules, such as large anions, proteins, remember they're in the blood. So their concentration goes up because they're losing water. When their concentration goes up, they will retake the water back. So they force the water come back. So that the scientists measure it. You don't have to memorize the individual number. You have to memorize the net. So the 32 mercury pressure that is coming back in, and then capsular space means any liquid, they put pressure on wherever they are. So they're in the capsular space, they also send back, and that's approximately 18. Again, I know this is a little bit of physics involved, so I'm mentioning it right now. If you don't need to know the detail. I mentioned it. What you need to know, the net filtration pressure is 10. That is helping the filtration to take place easily. Is there an additional help to keep that? Because remember, our body pressure is not fixed. They go ups and downs throughout the day. It's not that you have a hypertension, but it's not 120 fixed or 110 fixed. So they go, I'm just talking about the systolic, same thing with the diastolic, they go up and down. Like right now, maybe 110, 2 seconds later, 120, maybe 130, because based on your stress and other factors. Okay. So if this changes, these numbers should also change, but that actually does not happen because we have control on these capillaries. So we have control on these capillaries. So there are two ways we can control that so that they can overall keep a constant filtration pressure. That control I'm going to talk to you about a little bit later, that what makes that control so that net filtration pressure will remain around that 10 anyway. This we talked about that day. I think we did that math. This formula you don't have to memorize. You just need to know. It's not a very hard formula anyway. You just need to know glomerular filtration happens 125 milliliter per minute. Should be smaller, or 180 liter per day in case of male. In case of female, a little bit low. You don't have to memorize individual numbers. Just memorize that we absorb 99% back. We reabsorb them. So we just let 1 or 2 liters of urine per day. So we reabsorb 178, like 168, depending on the sex. We absorb them back. The rest, a little bit, we let it go. That's the urine. So where this reabsorption taking place, it is taking place in this area. I'm going to go back to those 2 slides later on. So it is taking place in this PCT and in the DCT. So in the PCT, proximal convoluted tubule, when you read the book, you'll see there's like a paragraph after paragraph. Just know these. I can't make it very condensed. So water, and go to the higher numbers, so it makes easier. Water, like they 70% absorbed in the PCT. So that's a cup. That is the PCT. That's a loop of Henle, and that's the DCT. So in this area, the water absorbs 70%, 60 to 70%. Same thing with the salt, 60 to 70%. Organic substances like glucose, amino acids, vitamins, like 99 to 100% absorbed right there. They just come back to the blood. Job is done. Loop of Henle. Loop of Henle has 2 loops, descending, ascending. In the descending, most water comes back, like another 30%, 25 to 28% water comes back right there. And in the ascending part, the salt comes out. So another 20, 25% salt comes out. And then, so think about 70% water here. So 25% water here. So 95 is gone, then 5% water remaining. That is taking place, that control taking place in the distal convoluted tubule, as well as collecting that. So they come out there. But if you pay attention to this particular slide, you will see there like a solid line they're showing here, and this is like a dotted line. Why this is different? The difference is this: 5% is a lot. Remember, 5% of 180. is a lot, like 10 liters approximately. So that much water. What that is? So the difference between these two, difference between this part and this is this side right here, when the PCT and the loop of Henle, that's called obligatory water reabsorption, meaning it's going to come back. You don't have to do anything. Osmosis, high concentration, high amount of water to low amount of water. So they will just come back from high to low. Water, not salt, water. So a lot of water here, less water here, boom, they will come back. So 95% takes place, that body does not need to do anything, and you have no control on this, obligatory. But this 5%, you have an additional control. So if you are hydrated, if your body has a lot of water, you say, let the water go. That means you urinate more. If you are thirsty, you don't have that much water in the body, you want to keep a lot of water, so your urine becomes concentrated. How you control it? That's called facultative water reabsorption. That's the hormone in posterior pituitary. We release that hormone called antidiuretic hormone, ADH. So this part is controlled by the hormone ADH. So ADH regulated permeability. So ADH is controlling the DCT and the collecting duct area to absorb more water or to let it go. So if the ADH is not there, it'll let it go. So I would give you a simple thing like more ADH equal more reabsorption equal less or concentrated urine. You have to understand this. More ADH equal more retained, more reabsorption in the body, less urine, so concentrated urine. That's why if you pay attention, you will see the color of the urine changes based on the concentration. So if you are hydrated, you are drinking plenty of water, your urine will be a little bit light straw color, like amber color we call it. But when you are not drinking, you're very thirsty, you will turn your urine colors like a darker side of yellow. The reason is you have less water coming out. Why? That control, the ADH controlling that. That is why that 5% control in this area is called facultative water reabsorption. Means we control them. Obligatory, you have no control. So that's how they take the fluid back. And now, when you study the book, pay attention to this one. There'll be a lot of word called co-transport, this detail. You don't just read them once. For this particular class, I'm not going into the individual cell level detail, like which cell does which way, what kind of co-transportation involved, because there is like active transport, passive transport, facilitated diffusion, co-transport, many things involved. We don't want detail. We want this, what absorb where. Let's go back a little bit again. So right here that we talked about the pressure, meaning the afferent arteriole bringing in here, the net filtration pressure is 10 millimeters of mercury, the efferent going out, the filtrate is right here. We have 180 liters of filtrate, which contain a lot of good stuff, a lot of bad stuff. We let the good stuff come back, and we learn that along with the water. And we let the bad stuff continue secreting from the body. Now, what controls this blood pressure? As I said, throughout the day, the blood pressure is not same. So this is their control. This is their control. We have two types of control. One is called myogenic control. One is feedback mechanism. Myo, the word means muscle, basically. So what basically it is, like arteriole, they have a lot of muscles. So those smooth muscle, when they constrict and relax, they control the blood flow. Basically, it is if you think about the pipeline, if you squeeze a pipeline and if you relax a pipeline, and thereby you can control the flow of water, same thing. By doing this, you are controlling the net filtration pressure. So although your body, blood pressure is fluctuating a little bit, just by doing constriction and relaxation, you can do this. So when the blood pressure falls, the afferent artery relaxes, so more blood comes in. So more blood comes in, the urine still can filter. When the blood pressure rises, the afferent artery squeeze in so that less blood comes in, so prevent the blood flow. By controlling the blood flow, by squeezing and relaxing, they can control the blood pressure, that myogenic mechanism. But there is a feedback mechanism in place also. Feedback mechanism is done through the hormone renin, or some book will call them enzyme renin. And then there is another cell also. Now, what they do it through a bunch of cells together in that area called juxtaglomerular apparatus. So near the glomerulus, juxtaglomerular apparatus. Now, juxtaglomerular apparatus, in that apparatus, there are three special kind of cells: macula densa, juxtaglomerular cells, and mesangial cells. So forget about these two, just focus on the third one first, mesangial cells. They're basically in the cleft between afferent and efferent arterioles, and among the capillaries of the glomerulus, they're sitting on top of them. So let's say this is the blood vessel, this is the blood vessels, this pipeline that's sitting right here. These cells squeeze the blood vessels, so that they can literally squeeze the blood vessel, relax the blood vessel, squeeze the. So by doing that, maintaining the blood pressure, that's called mesangial cells. So in this picture, like these cells right there, you know, this purple color, that's a mesangial cells. So those cells are squeezing the blood capillary and this glomerular filtration area to control the blood pressure. But there are two other varieties: macula densa, and juxtaglomerular cells. So where are they? Okay. Now, in order to understand, let's go back to the nephron structure first. So when you study nephron, any nephron structure right here, to understand, we put the cup here. That's a filtration cup. That's convoluted tubule is called PCT. This is the descending loop. That's the ascending loop, so loop of Henle, and that's DCT, brings it to the collecting duct. Simple structure. But in reality, that's not the case. In reality, the loop of Henle does not bring the DCT away, actually. It is technically away, meaning it is the leader pipeline, but the pipeline, this portion, passes through this area. So in true structure will be, if you flip that structure, meaning this loop of Henle will go up, and then the DCT will come here before they go back to the collecting duct. What does that mean? That means you pay attention right here. That's the nephron. So there's a blood vessel going on. That's the loop of Henle. Not loop of Henle. That's the renal corpuscle. So that's renal corpuscle. This is the PCT. Then PCT goes here, proximal convoluted tubule, makes a loop. Then loop brings to DCT. DCT come close to the. I think I'm looking at the wrong way. Hang on 1 second. Let me look at the bigger structure right here. My eyesight are bad. Okay, that's the PCT. That's back of the. This is the cup. That's the blood vessel. So opposite side of the blood vessel is the PCT. So this is the PCT. That's the convolution. That is where the loop. This is the DCT, and that goes to the collecting duct. So see, the point I want to make is DCT, the DCT and the afferent and efferent arteriole come close together. That's the point I want to make. The DCT and the afferent and efferent arteriole come close together. Look at this also. That's a blood vessel, afferent, wider, efferent comes out. That's a renal corpuscle, that's PCT. PCT going this way, make a loop, huge long loop, then DCT, and then it goes to the collecting duct. Again, look at the junction. Right here, DCT come close to the efferent and afferent arteriole. This is afferent, this is efferent arteriole. So that structure, when you blow it up, it looks like this. It looks like this. So that's the cup. Afferent going in, efferent comes out, PCT on this side, DCT on this side. So the DCT comes close to the junction. The cells in DCT is normally cuboidal cells without microvilli, but look at these cells, they become tall. These tall specialized cells, when they come close to that junction, are called macula densa. This population of cells are called macula densa. That's a macula densa, that population of cells. And these population of cells, which are sitting on the smooth muscles of the afferent arteriole, are called juxtaglomerular cells. These cells, all of them together, create apparatus called juxtaglomerular apparatus. What they do, they release a hormone called renin. They release the juxtaglomerular hormone, renin, released by this juxtaglomerular cell when the blood pressure drops dramatically. So they're keeping an eye, the DCT is keeping an eye, and then they release the renin. Now, renin can act as an enzyme. That's why some books call it enzyme, some books call it hormone. Now, 3 steps, you need to know this. Renin, what they do, the first step, they convert a protein called angiotensinogen. Angio means blood vessel. That's why people do angiogram. So angiotensinogen, there's something to do with the blood vessels. So it's a protein, they convert them into angiotensin 1. That's the renin job. So when the blood pressure drops, kidney juxtaglomerular cells secrete renin, they go to the blood, they convert angiotensinogen, which is in the blood, to angiotensin 1. That angiotensin 1 will be converted to angiotensin 2. Who does that job? There is an enzyme called angiotensin converting enzyme, SEE. Who produced that? Lungs and the kidneys. They can make that enzyme. So in that enzyme, they convert angiotensin 1 to angiotensin 2. That becomes like an active hormone. So angiotensinogen and angiotensin 1, they're inactive. So angiotensin 2 is active. So what they do, they go to the adrenal gland. Now, adrenal gland, we learned that they have a cortex and a medulla. So in the cortex, they go there. In the corticular region, this adrenal gland goes. And the angiotensin 2 goes. And in that response, they release a hormone called aldosterone. So look at these structures. So angiotensinogen, which is produced by the liver, they're in the blood. Renin comes from the kidney. Juxtaglomerular cells release them. They convert them to angiotensin 1. Angiotensin 1, the lung produce enzyme called angiotensin converting enzyme, produce angiotensin 2. That angiotensin 2 comes to the adrenal gland, release aldosterone. Aldosterone goes to that DCT area, that collecting duct area. So aldosterone goes back to the DCT and the collecting duct area. These are the pump. They are controlled by aldosterone. Aldosterone regulated pump. The letter A represents aldosterone. And that, what they do, they're bringing a sodium ion. So their aldosterone, remember in the hormone chapter, you learned that aldosterone is responsible for sodium metabolism. This is what actually happens. So this pump, what they do, they bring in sodium. So when the sodium comes, water follows. Remember, water follows salt. So when the sodium comes, water comes together. So water follows the salt. So when water comes back up, blood volume increases, blood pressure goes back to the normal. That's how body control the blood pressure. That is why it's called renin angiotensin aldosterone mechanism, RA mechanism, RAA. So it's like a few steps, so the blood pressure increases to normal. That's why you know this, like when doctors recommend don't take too much salt when you have hypertension. Why? Because if you have too much salt, more retention, more water will come back, blood volume goes up, blood pressure goes up. That's the mechanism we just studied. So that's it. That is how actually we make urine, and we control all the water reabsorption. The last part, which I already mentioned, ureter, urinary bladder, urethra. Ureter, these are the muscular tubes. They bring the urine from the kidney to the bladder. Urinary bladder, a muscular organ which temporarily store the urine. Urethra, another tube or pipeline which extend from the urinary bladder to the outside. Two sphincter that control the micturition reflex. One inside is called internal sphincter. One outside called external sphincter. As discussed earlier, internal sphincter is smooth muscle controlled by the central nervous system. External sphincter is the skeletal muscle. They also need a nervous system influence, but that's a skeletal muscle because you have conscious control on that particular urethra, particular sphincter of the urethra. That's how we control the excretion of the urine outside. This is the histological structure of the ureter. We mentioned that in the lab class that you should know the look of this, how they look like. And that is why, I'm going to mention one thing. This ureter has a tremendous amount of pain receptors. So the kidney stone, like you heard about, obviously people have severe pain, colic stone, because the stone gets caught between this star-shaped internal area, and that cause tremendous amount. It's not like a simple pipeline. It has an inside, it's like this, star shape, very typical diagram. So that's how the urine is passing through them, like the ureter to the urinary bladder, and urethra goes out.